All right. So thank you, Thorsten. Thank you for having me today. It's a pleasure to talk to you today about what we do in the NeuroCytolab in Marseille to look at the axonal cytoskeleton down to the nanoscale. So these are neurons. So this is what we study. And as you can see, these neurons have a very diverse and striking morphology. So you have some that are very branched, very large, some that are very regular, some that are very irregular. So really the mystery of that is how do they build, maintain, and transform this very unique architecture that is, of course, necessary for their function in the brain. And the neurons we are looking at are simpler than that. We are looking at them in culture. We are looking at the cell biology side of things. So we use hippocampal neurons in culture. And you can see them growing in this long-term movie for about 16 hours. And you see that they grow small dendrites and a longer axon. And this is their main property, which is that they are polarized. So they have the cell body and dendrites on the left here. I'll try to get the pointer just yes, okay. The cell body and the dendrites on the left and the axon on the right that is longer and that connects the cells. And of course, this polarity is even more extreme than that because the axon is actually much longer usually than what this cartoon is showing. And I drew a neuron here, which would be a motoneuron that has a 1 meter axon and to scale the cell body would be here. And here you would have only 20 centimeters of its 1 meter of axon described. So you can see the challenges of building this very thin and long tube and bringing the cellular components there and everything. And of course, the polarity reflects the function of the neuron, with the cell body and the dendrite that receive the inputs and the axon that generates the action potential that propagates to target cells. And the axon itself, the thin and long tube, is compartmented into subcompartments, such as the axon initial segment, here in green, the presynaptic boutons in red, where the axon contacts the target cells, and in growing neurons, the growth cones at the tip of the axon. And this is what we are studying in my lab. We're trying to understand how these subcompartments are built, maintained, and transformed throughout the life of the neuron. And of course, this is the cytoskeleton and unique structure of the cytoskeleton that support this unique organization. And so most of what we try to do is to map the structures of the cytoskeleton inside the axon to understand the functions, the physiological function of neurons. And if you want to map the cytoskeletal structure, we have a range of techniques that allow us to see these structures. And classically, this has been done by electron microscopy. I mean, even before that, it was done just by Ramon y Cajal, just looking in a microscope and drawing it, like you can see here. But after that, electron microscopy in the second half of the 20th century was used to define the different structures, such as the microtubules or the actin, and different types of EM would give you different types of details about the structures. But the thing is that this does not identify them at the molecular level. You see structure, but you are not sure what they are. And so what is able to identify them is fluorescence, immunofluorescence. So you label something, microtubule or actin, and you know that you're looking at a microtubule or at an actin or a structure. And these are at the top here is the first example I could find of immunolabeling of cytoskeletal components in a neuronal cell back to 1978. And at the bottom is the classic way of doing it these days, which is using phalloidin for actin and antibodies for microtubule here in hippocampal neurons. And the problem with fluorescence microscopy is that it is limited in its resolution, in the amount of details you can get. It is limited by the diffraction of light. And even if you have the best microscope available for fluorescence microscopy, the best detail you can get is the size of what is called the point spread function, PSF, which is about 200 nanometer in width and 500 nanometer in height. And this limits the maximum resolvable detail. If you look at something that is super small, like a single fluorophore that is about 2 nanometers in size, you will see a spot that is the size of your PSF, 200 nanometer in diameter. And if you look at a neuron that is labeled for different components, you see that in addition to being pixelated, this image also is blurry because, for example, the bassoon, which contains the presynapse components, are single compartments that are smaller than the smallest detail you can resolve. And so you just see them as something that is about the size of the point spec function. Fortunately, for about 20 years now, we have new techniques that allow us to look with more details using fluorescence microscopy, and all these techniques are collectively termed super resolution microscopy. And there are a lot of different techniques like STORM, STED, PAINT, lots of acronyms. I won't get into all the details. I will give you a few pointers for the ones we are using. The one technique that we use can be collectively termed single molecule localization microscopy. And the idea is that you are looking at the details by localizing fluorophores one by one. And why can you get more details with that? So here on the left, you have a classical staining, microtubules in a fibroblast. And you see that they are blurry because of this diffraction limit and the PSF. But if you transform this staining into a very, very sparse staining where only a few of the labeling fluorophores are active, you see that each of these fluorophores, you can localize them with a better precision than the size of the spot by just fitting the center, and then start to make a graph where you will put each of these localizations on a graph. Of course, if you do that only for one image, you will have like 6 or 7 fluorophores that will not give you the final image. But if you transform your sample into a blinking sample, where these will blink on and off randomly, and at any given time you will have only a very small fraction that are active, you can do that for thousands and tens of thousands of frames. And you accumulate all this fitted localization, and in the end, you draw all of them on a graph, and the final image is your single molecule localization microscopy image, which has a resolution about 10 times better than the PSF size. So you get down to 20 nanometers rather than 200. And this is what is illustrated here. Your sample is blinking, you fit the position of each fluorophore, and then you have another image, and you fit again, and you do that for 60,000 frames, typically. And then you accumulate all these localizations, and you get the final image that is much better result than the fluorescence diffraction limited image. So 2 of the flavors we use to do that are called STORM and DNA PAINT. Both are SMLM techniques. STORM, you use a classic immunolabeled sample, and you put it in specific conditions with a reducing agent and a high laser power that makes them go to the dark state, and only a few of them are blinking. And the other is DNA PAINT, where instead of having fluorophore on the secondary antibody, you put a small DNA strand that is called the handle, and in the solution during imaging, you put an imager strand that has a fluorophore, and this is the transient interaction of the imager strand with the docking strand that will transiently immobilize the fluorophores and make the blinking. That you will just fit both techniques. You will use the same kind of processing, fitting all the single molecule blinking events to reconstruct the final image. So how do we apply that to the axonal cytoskeleton? And this example comes from 2013, when the lab of Gerwei Wang with Kexu started to look at actin along axons using STORM. And what they found when they looked at the diffraction limited image was not very interesting. You see that this axon is labeled and it's mostly uniform with the actin staining. But the STORM image is actually much more interesting, because this uniformity, if you go beyond the diffraction limit, actually hides a very striking assembly of periodic rings. So you see the rings of actin along the axon, and they are regularly spaced every 190 nanometers. And this 190 nanometers is the reason why it appears uniform at the diffraction limited level, because it is just under the diffraction limit. So after blurring, it just looks uniform. But it's actually very precise succession of rings every 190 nanometers. And why is this distance 190 nanometers? This is because between each of these actin rings, you have a single layer of spectrin tetramers. So spectrin tetramers have a length of 190 nanometers, 2 alpha and 2 beta spectrin, and they are spacing one ring from the other. They are connected to 2 rings, and in between each actin ring, you have a layer of spectrin tetramers that define this regular distance of 190 nanometers. And this is what is called the membrane-associated periodic scaffold, or MPS, that is present along axons. And you can see here on this image that the axon has this very striking periodic scaffold. The dendrites sometimes have it, but usually it's much less visible. And you see that if you wait a lot of time, they start developing something partially, but this is really more present along the axon and much sooner in the development. And the important thing is that this is a fundamental constitutive element of the axon. Like all axons in all types of neurons, in all organisms where it has been looked for, present this striking periodicity of the actin spectrin scaffold. And it's really, for me, one of the most important structures that has been discovered by super resolution microscopy, because it is entirely new and was never observed before. It's not like super resolution microscopy would give more details on something we already knew. It's really discovering a completely new structure, like discovering the Golgi apparatus or the mitochondria. And so this MPS is obviously very important because it's ubiquitously present in all axons of all neurons in all animals. And the question is, what are the functions of this scaffold? And the first one that was hypothesized was that this would endow axons with more mechanical resistance. And the model is the vacuum cleaner hose model. So a vacuum cleaner hose is very flexible, but also very resistant because it has these rigid rings connected by flexible plastic. And so you can really change its shape. And the idea is that the axons need that because, of course, not in your brain, but in your peripheral nervous system. All your axons must withstand a lot of stress when you are moving and when the body is growing. And so this is also shown by, to the right here, the C. elegans worm that are lacking the spectrin, don't have this scaffold, and they show during their life the axon break because of the movement of the animals. So it's a way to show that really the scaffold is giving mechanical resistance to the axons. And this is the kind of images we do in the lab now. So for about 10 years now, we've been working on trying to understand this scaffold and its organization and function. So we have made advances and found new partners and refined the organization. You see, these images are two-color SMLM images that are hybridization of storm and paint, where you see the spectrin and the actin rings that alternate. This is beta-4 spectrin in the beginning of the axon, the initial segment in yellow. And in the distal part, the axon contains tetramers of spectrin that have beta-2 spectrin. So alpha-2 and beta-4 in the initial segment and alpha-2 and beta-2 in orange in the distal axon. And so if we do this even better, we can do 3D imaging and make nice reconstruction and roller coast along single axons. So you see this culture here and you have all these little zooms. And this is what is on the right. And you see the lumen of each of these axons and the periodicity of the spectrins. And we can ride along axons and see their details. And this gives you an idea of the precision of the technique because all these lumens of axons are smaller than 500 nanometers. So this is much better than a confocal or classical fluorescence imaging. Nonetheless, these are not the best, these are not the definitive images because you don't see the ultrastructure. You don't see really individual filament or individual spectrin tetramers. So there's a limit to what you can say about the structure and the organization of the scaffold that you cannot reach with any optical route technique. And this is why some time ago we teamed up with Stefan Vassilopoulos at the Myology Institute in Paris, who is a specialist of a technique called platinum replica electron microscopy. This technique allows you to see the single elements of the cytoskeleton that is associated with the plasma membrane. And it does that by you start with a cultured cell and you unroof it with a sonicator, the same sonicator you use to do membrane preparation. And you are left with the ventral part of the cell that is attached to the glass. And then you make a carbon replica of this and you examine it with an electron microscope. And so you see the inside of the plasma membrane and all the cytoskeleton that is attached to it, like these actin filaments or these clathrin network, clathrin plaques here, which are one of the objects Stefan is studying. So we thought, okay, the actin rings and the spectrin mesh are submembrane in the axon. So if we can unroof axon, we might be able to see by this electron microscopy technique the ultrastructure of the MPS. And at the time, nobody has ever seen the MPS with electron microscopy, which even some people were doubtful it was existing because they said, why has the electron microscopy never been able to see it? So we managed to unroof neurons, and we could unroof the cell body and the proximal neurites, including the axon, so that was good. And if you look at this image, EM image of the replica, you see these are the parts that are unroofed. The bright white ones are where it's not unroofed, so the distal dendrites and axon are very difficult to unroof. But we can see the beginning of the axon here in yellow. And if we zoom, we see that inside, so this is the plasma membrane, and you see the microtubules here that have very dense bundles that are called fascicles that are specific to the axon. And you see that the plasma membrane doesn't look like the previous image I showed you. It is completely covered with a mesh. And if you zoom on this mesh, you see these vertical magenta filaments. And these filaments are actin filaments. And actually, if you zoom further, there are two actin filaments braided together. And you see that the distance between these vertical filaments is about 200 nanometers, and they are connected by this mesh. And so we said, okay, maybe these are the actin rings and the connecting mesh are the spectrin. And we demonstrated that by doing immunostaining, immunogold staining, and things like that. But the definitive demonstration that these actin braids, as we call them, because they are a braid of two filaments, and yes, I know a braid is supposed to have 3 filaments because I have daughters, but still it's just 2 filaments. But these actin braids, we needed to demonstrate that they were really the same object that we see in super resolution microscopy. So we did a correlative approach. So we unroofed neurons and we stained them with phalloidin. So you see the actin rings that are seen by storm here. So these are the beginning of this axon. And if you zoom, these are these vertical lines here are the actin rings. And the distance between two of them is about 200 nanometers. And then after that, I would make a circle with the diamond tip objective and send the cover slip to Stéphane in Paris, who would do the platinum replica of this exact same neuron and look at, and find it in the electron microscopy. And then we would align the two images. And that was possible because the two techniques have a very high resolution. So you see it clicking when they're aligned. And when you do that, so this is the EM replica, and you see that the axon is split open with the microtubule inside. And when you have the mesh here, and you see these vertical braids, but you don't see them all along, like they disappear in the mesh, on and in and out the mesh. But when you superimpose the storm image, it's very clear that each of these vertical filaments, the braids on the prem image, are indeed the rings on the storm image. And so this is the first and still only, to my knowledge, EM visualization of the MPS. And we could see that they are not short filaments, but long filaments braided together, which has, of course, important consequence for the organization and stability and mechanical properties of the scaffold. And while we were doing that, Stéphane, who is a specialist of platinum replica electron microscopy and clathrin-coated pit endocytosis, kept saying me, but look, there is the mesh, there are the actin braid, but there are also this structure here, and this is a clathrin-coated pit. So it's very interesting that nobody has described clathrin-coated pits or endocytosis from the axon shaft, and maybe we should look at that. So that's what we did. And this was the work of Florian Werner, his PhD in my lab, and Satish Mukherjee, postdoc in the lab of Stéphane. And if by that time we had a new microscope, a structured resolution microscopy microscope, that would allow us to see the periodicity of the spectrin, as you can see here in green, and very easy multicolor imaging, so we could see individual clathrin-coated pits within the spectrin mesh along the proximal axon of this neuron. And here it's an intact neuron, here it's an unroofed neuron, and in the unroofed neuron, it's even clearer that all the little spots, the clathrin-coated pits in magenta, they are exactly in holes of the spectrin mesh. So every of this clathrin pit is a little hole of the spectrin mesh that allows it to access the plasma membrane. If you do an averaging of hundreds of these clathrin-coated pits, you see that the spectrin will make a little hole. So you really have each of the clathrin pit is encased in the mesh of the spectrin. And of course, if you do platinum replica EM, you see these holes with a much better resolution. And you can see it here. Each of these holes is what we call a clearing. So looking at these images, and in particular, so you see different stages of clathrin-coated pit formation inside this circular structure that are devoid of the mesh. You see here it's the bare plasma membrane, and you see that inside you can find nascent clathrin-coated pits or fully assembled or even two of them. And looking at this image of the archetypal structure, we saw, it made us think about the witch in the forest that lives in this house that is a clathrin-coated pit, and you have this little path to go there, and it's the clearing of the forest. So that's why we called it a clearing. So the clearing is this circular structure of about 300 nanometers that is devoid of the spectrin mesh, and in the center you have a clathrin-coated pit. And the path here is actually an actin filament. And every time you will see an actin filament connecting from the periphery to the center of the clearing and touching the clathrin-coated pit. And so we also used our correlative workflow to localize actin and spectrin, and we can demonstrate that indeed the path here is made of actin, and that the periphery of the clearing is usually enriched in actin as compared to spectrin. So this is how it looks like, this structure. It's a round structure that allows access to the plasma membrane and the formation of clathrin-coated pits in the very dense mesh that covers everywhere else in the axon. And of course, you can imagine that if the axon is covered with this, it will be very difficult to do endocytosis. So this allows to have endocytosis along the axon. But does it really happen all the time? And this is actually the surprise when we did that, is that when we started looking at it live, so we did that with structured illumination microscopy, which is based on using a structured light and several phases, so 15 images or 9 images, and after that, with a mathematical processing, we can retrieve a 2 times better resolution. So we can do live, but we have 120 nanometer resolution compared to stored, only fixed, but 20 nanometers. And it was shown that the NPS could be visualized by structured illumination microscopy back in 2017 by the lab of Andreas Prokop. So we did that, and we looked at the spectrin and the clathrin together by 2-color turf sim, and we could see the clathrin-coated pits and the spectrin, and each of the clathrin-coated pits encased in a little hole of the spectrin. But the surprise comes from the fact that these clathrin-coated pits just stay there for the whole movie. 20 minutes, this movie is 20 minutes, and the same clathrin-coated pit can be seen for the whole duration, which is super long-lived compared to a classic clathrin-coated pit. If you look at this is a glia next to the neuron, and you see everything is blinking because the clathrin-coated pit appear and disappear constantly. So the stability, and you can see it here, the kymograph showing that these clathrin-coated pits are there from the beginning to the end of the movie. Of course, the spectrin scaffold is also very stable during this time. And so in all of the spectrin, you have a stable clathrin-coated pit. So this structure, the clearing, does not make a place where you will have active endocytosis all the time with clathrin-coated pit appearing and disappearing. It makes a hole so that you can have a clathrin-coated pit that is then frozen, waiting for something to happen. And this makes sense because neurons don't need to endocytose everything all the time. And so we could demonstrate, I will not give you all the detail, but if we give the right stimulus, which is a long-term depression-like stimulus, NMDA treatment, then we could trigger the endocytosis from this clathrin-coated pit. And so the clathrin-coated pit would be frozen, stalled, and after stimulation and triggered endocytosis, the actin would start to polymerize into the clearing, and we think it's from these little actin filaments that we see always. Like from that, you would have a polymerization of a lot of branched actin that would help finish the endocytosis and cut off the clathrin-coated pit so that it can go inside the cell. And so this is a new way of doing regulated endocytosis that we published last year or 2 years ago. And I would like now to talk about the converse process. I told you about endocytosis, but what about exocytosis? And this is a work that is being finalized by my postdoc, Teresa Wissner. And the idea is that you can have exocytosis and endocytosis at the presynapse where you don't have the MPS. But in the shaft where you have the MPS, we have shown that endocytosis can happen in very specific cases within clearings and in a regulated manner. But how can you have exocytosis and how does it work? Because you also have to overcome this mesh if you want to get something out along the shaft. So what Teresa has been doing was to use fluorine, which is a fluorescent protein that is dark in the acidic compartment inside, and when it's exocytosed, it becomes fluorescent. So if you bleach the surface protein and then you make a movie, you will see flashes that are exocytic events. And this is the only way to properly see exocytosis. And of course, it means that you have to do live cell imaging. You cannot do like endocytosis, just look for clathrin-coated pit on a fixed cell. You have to do live cell imaging. So of course, this makes it a bit more complicated. But if we make these movies and we label where are the synapses, you see these flashes and they can be at a presynapse, which is labeled in magenta by the synaptofluorine M cherry, or along the axon shaft outside of presynapses. So exocytosis can happen in synapse and outside of presynapse. Of course, this is a very low occurrence. This is unstimulated neurons, so the amount of it is about 2 events per micrometer square per hour. It's not like, synaptic exocytosis. If you stimulate, you will have lots of events in a short amount of time. And actually, these 2 events, because people usually show you this kind of very small region of interest and you don't know what you're looking at, so I'm trying to explain here. This is this part of this axon that is transfected with the synaptic marker, magenta, showing you all the synapses, and the fluorine that is in blue. And every time on the kymograph you see a vertical, it means that it's there from beginning to the end. And if something is appearing or disappearing means it's endocytosis, appearing is exocytosis of this event, or this event here. This one is outside of the synapse. These 2 are inside synapses. And Teresa started mapping all the exocytic events along axons using these fluorine constructs, labeling if they were at the initial segment, along the proximal axon or the distal axon, and comparing them to the cell body and dendrites. And you see a single movie where you have these kind of events at each of these compartments. And here is like you see these flashes. So in the cell body, you have a lot more basal fluorescence, so it's more difficult to see, but you see them. And then quantifying their occurrence and their frequency. And what was really striking is that the initial segment, which is supposedly a very stable compartment, completely covered with spectrin, has 2 times more exocytosis on average by surface than the proximal or the distal axon. So it seems to be a preferential place for exocytosis. And next, we wanted to know, okay, so if there is exocytosis along the axon, does it need also a hole? It needs to access the plasma membrane. So how does it work? Do you have holes in the spectrin that allow for the exocytosis to occur? And to do that, Teresa set up a correlative approach of live cell imaging and storm. So we're not doing correlative storm and FEM like we did for endocytosis. We do live cell and then we fix and we do storm. And that works like that. You have the live cell imaging movie that gives you here 2 examples of flashes of exocytosis along this axon. And then after fixation, she will do a spectrin staining and the same axon segment, and you see the periodicity of the spectrin. And to see it better, we teamed up with Ricardo Enriquez in Lisbon, who is developing a lot of image processing approaches. And he developed something called shred that allow us to see exactly where is the spectrin and where it is periodic. So we could define, okay, this is where is the spectrin and this is where we have holes. And after doing that, we could superimpose the live cell movie and then look at where exactly was the exocytosis happening compared to the spectrin organization. And when we did that, we could see that actually these were all in the spectrin. But when we did that with 2 colors, by doing storm for spectrin and clathrin, we could not only see that the endocytosis was happening in holes of the spectrin mesh, that is in the area where you don't have the green rings, but also in holes that don't have clathrin-coated pits, which are here in magenta. So you see here holes that are clathrin-coated pits, and here it's a hole that is empty, and this is where the exocytosis will occur. So it seems to imply that there are two types of holes in along the axon. One that are used for endocytosis, the clearings that we were able to visualize down to the precision of electron microscopy, and the exocytic holes. We don't think they are clearings. There might be different type of structure. We cannot do really platinum replica EM because we cannot do correlative live cell to prem because the prem is not efficient enough to target a given axon that we have looked at by live cell imaging. But we think that these are different structure or holes that don't have clathrin-coated. And you can see a lot more details about this in our recent preprint that is on bio. And that's it, I think. So thank you for listening. And this is the team. And the work I showed you has been done for the endocytosis by Florian Werner, who left the lab a couple of months ago, and the postdoc work about exocytosis by Teresa Wissner. These are our collaborators, founders, and partners. Thank you very much.